Good morning, everybody. Welcome to my office. We've been kicked out of the seminar room because they're getting ready to remodel Harris Hall on campus, so they're moving all the faculty that are located in that building in other spots around the campus, and so they have absconded with our seminar room. So for the time being, I have moved the Integrity card into my office, and so welcome. Today, we're going to be talking about acid-base balance problems. So many nursing students have for years complained they didn't understand how to do those, they're so hard, and I'm here to tell you it's easy. When you are working acid-base balance problems, what you're trying to do is take a set of lab values that you have been given from a patient, and you're looking at the homeostatic ranges of those particular lab values, and you're trying to determine whether a patient is in acidosis or alkalosis, and if so, whether their problem tends to be more of a respiratory problem or a metabolic problem. And I've heard a lot of different, memory techniques, mnemonic devices and the like, but I'm a believer in why not understand it? If you understand what acid-base balance problems are and what it really means, and follow a very easy 4-step method, and science is a methodical thing. We have step-by-step ways of doing things, and I'm telling you, if you will understand what I'm getting ready to teach you today and follow the step-by-step method I'm going to teach you, it's fail-safe. You will get it right every time. You want to do that, don't you? Okay. Let's see, before I get into this lesson, I want to make sure that you go online and that you print off the 4-step method. I think I have put it in your Unit 3 case study folder. It's called Understanding Acid-Base Balance in 4 Easy Steps. It's going to look like it's ancient. Well, it happens to be over 20 years old. I went to an A&P Human Anatomy and Physiology Society conference a long time ago and picked this up from one of the lead instructors at that conference. And he was teaching this way, and it still works today. So I'm going to teach you an old method, again, that is a good method. So without further ado, let's do a little bit of talking about first the rationale behind it and setting it up. The first thing is that the homeostatic ranges you're going to be interested in on this are typically taken from blood samples, and so we call them arterial blood chemistry, so the samples have to come from the arteries. A homeostatic range for the pH of arterial blood is, or any blood really, is 7.35 to 7.45. We're going to review pH in just a minute. The other value you're going to look at is the pressure in the arteries exerted by the gas carbon dioxide. And the pressure, it's a partial pressure. A partial pressure is just knowing that there's other gases and other things contributing to the pressure, but here is its part. So carbon dioxide as a gas contributes about 35 to 45 millimeters of mercury in a homeostatic range. And then you're also going to be looking at what we call the bicarb ion, the HCO3 ion. And it is measured in milliequivalents per liter. That's kind of a weird measurement. Just get used to the fact that that's the way the bicarb ion is measured. But its normal range is in the 22 to 26 milliequivalents per liter. All right. This is basic chemistry. How do you look at a pH scale? What is a pH? It's really the measure of hydrogen ion concentration and the release of the hydrogen ions in a solution, where when you have the highest amount of hydrogen ions released, you're going to have a more acidic condition. So the acids tend to, the whole scale. Oops, I forgot to get my pen. Let me go back. Hang on just a second. And I want to go back. there we are. Sorry about that. All right. So the pH scale ranges from 0 to 14, where 7 is neutral. And the numbering system from 0 to 7 to 14, the lower the number, the more hydrogen ions are released, and therefore the more acidic the solution is that you're testing. In this case, you're talking about blood. Up here, the less, I'm going to put fewer hydrogen ions are released. Sometimes people discuss this as there are more OH or hydroxyl ions released, but don't let that bother you. This is where it is on the more alkaline range, or basic. Okay? So, when you take a look at the homeostatic range, 7.35 to 7.45 for blood, then even though both of those, or that whole range is slightly alkaline, you consider which side of the range a patient's lab value is on if it's not within the range. So, if a patient's lab values are less than 7.35, they are considered to be in acidosis, because they're, ACIDO, I can't spell and talk at the same time. They're considered to be out of the range more toward the acidic side. They're releasing more hydrogen ions. If their pH value is out of the range higher than 7.45, then they're considered to be in alkalosis. And to be honest, you're not going to see any lab values way out or for any extended period of time, because what's going to happen? If this is a homeostatic situation and your body can't maintain this homeostatic situation, what will happen to you? You die. That's right. So your body has adjustments that it can make to try to keep the pH in this range. You might tolerate slightly out of the range for a shorter period of time, but typically you're going to have bad symptoms. As a result, you're going to be wound up in the doctor's office and ultimately in the hospital, where they're going to try to help you get that pH back in range. So let's go on to the next slide. We're going to look at the carbon dioxide levels of, in arterial, pressure of carbon dioxide in the arteries. Let's call it like that. We say P sub A. So that really should be written P and a little a down there. So it's the partial pressure of carbon dioxide in the arteries. That's what the A stands for. Whenever we are looking at that value, we're looking at what we call the respiratory parameter. You will understand more of this as we do some problems and you see what we mean. But think about it. Carbon dioxide as opposed to bicarbonate, what you're typically going to be doing that is involved in respiration is you're wanting to get rid of the carbon dioxide. Carbon dioxide is toxic in the body, so you want to get rid of this carbon dioxide, so you're going to breathe it out. We're going to talk just in a minute here where all this is coming from, but keep in mind that if you remember that you got to get rid of CO2 by breathing out, then it's going to be the respiratory parameter that's going to be considered. Well, then the next thing comes that ties back into the cardiovascular unit, and that is, how is most carbon dioxide carried in the blood? You remember? Most of it, 80% of CO2, binds to water in plasma and forms a substance called carbonic acid. 20% actually has can bind to hemoglobin, but most of it forms carbonic acid. So let me show you just a little formula. I'm not going to throw too much chemistry at you here, but I think y'all probably can see this. If you combine H2O plus CO2, you get a molecule H2CO3. See that? You combine H2's there, carbon, and then instead of 2 oxygens, now you have 3 oxygens. Well, this is carbonic acid, and it's a very unstable acid, so what it will do is quickly dissociate into 2 more stable ions. One is the hydrogen ion. I'm running out of room, so I'm going to put the other one, and HCO3, the bicarb ion. So this is considered the bicarb ion. So there's your hydrogen ion, which makes it an acid because carbonic acid dissociates very fast into hydrogen ions and bicarb ions. So using the blood PaCO2 range that was 35 to 45, here's what I want you to remember. If you have a lot of carbon dioxide in the blood, does that not stand to reason that you will have a higher amount of acid? You're going to be producing more carbonic acid because as your body needs to get rid of that CO2, 80% of it's going to combine with the water in the plasma and form carbonic acid. So the more carbon dioxide waste you have, the more acidic your blood might tend to be, right? So if this is a measure, 35 to 45 is a measure of, and I should go ahead and make sure you, what am I doing here? Oh, I know what I was going to do. Make sure that you remember that this is millimeters of mercury. This is a pressure thing. Anyway, if this is a measure of the CO2 levels, then the higher the number, the more CO2 you got, which equals the more acid or carbonic acid in the blood. And so the higher the number on the CO2, the more you're going to go into acidosis, right? Because you're going to have more acid, so the higher the CO2 number, the more into the acidosis range. So if you get outside of the 45, you're going to probably be in acidosis. All right, let's look then. What if the CO2 level is lower than 35 millimeters of mercury? If it's lower than 35, then you're not going to be producing as much carbonic acid, which means your body might be tending to go further over into the alkaline range. So if your CO2 level is out or lower than 35, then you're going to tend to be in alkalosis. All right, let's go to the next one. We're just setting it up. Give me time to set this up. I will repeat enough of this as we work problems that you will get it, okay? All right, now let's look at the other thing that we're going to be considering in our lab values, and that is the bicarb ion value. The bicarb ion is your metabolic parameter. This one is going to help you decide whether you have a metabolic problem, whereas PaCO2 was used to help you decide if you had a respiratory problem. This ion is the bicarb ion, and a bicarb ion acts as a buffer for the acid. Okay, it is a buffer. It is a base. So if your range is 22 to 26 milliequivalents per liter, the more bicarb ion you have, the higher the bicarb ion, the more what? The more base you have. So if you're out of range high on the bicarb ion, you've got more basic than you've got acid in the balance. So a high bicarb ion, higher than 26, is going to indicate what? That you might be going into alkalosis. Whereas if the 22, if you get lower than 22, then you don't have enough buffer for those acids. So a number lower than 22 on your buffer would tend to put you in acidosis. Now, before I go any further, I'm going to digress. And don't stand there, don't sit there at your computer going, oh, Miss Cottle, please stay on track, don't digress. I have enough trouble with this anyway. But you know what? For you to really understand this, you've got to see the big picture. And so I want to take just a moment to make sure you understand how involved all the systems are in maintaining pH. For instance, you're going to also be studying this unit, if you haven't already addressed it, digestion. And the digestive tract is based on the ability to break down the foods that we take into our body into an absorbable level. And in order to do that, we have to activate enzymes. For enzymes to be activated in our digestive tract, one of the the parameters that helps activate it happens to do with pH. And I know you guys know enough to know that your stomach has a really unusual pH. What is it? You all know people talk about having acid indigestion, right? So the stomach is producing hydrochloric acid. Now, normally it won't produce very much of this unless you take in a lot of protein in your diet. So as you take food that's got protein in it, we begin the breakdown of proteins in the stomach by activating an enzyme whose precursor, whose inactive state, is called pepsinogen. But when hydrochloric acid is secreted by certain cells in the stomach, the pH is going to drop, and what that does is convert pepsinogen into pepsin. And so now that enzyme can start to begin the breakdown of proteins. All right, so we've got this really low pH. In fact, digestion, the hydrochloric acid can make your pH in your stomach go down to about 2. That's really acidic. Most acidic thing in our body, as far as I know. But when you're through with stomach digestion and you've got to take that food on down into the duodenum and jejunum, where other enzymes are going to be involved, the rest of the enzymes down the track require more alkaline environment to be active. So they're actually not active or inactive if you've got too much acid. So what you have to do as the food is moving on down into the duodenum is you've got to secrete a bicarbonate. Remember what that is? It's a buffer. It is a base from the pancreas. So the pancreas is going to be contributing a bicarb ion into the digestive tract at the duodenum to neutralize the acids coming from the stomach and to activate the enzymes that are further down. So there you go. The digestive system is important. So let's take it one step further to continue to tie all these systems together. If you think about your 4 basic organic compounds in the food we eat, we've got carbohydrates, proteins, lipids, and nucleic acids. Okay? We're going to focus right now on the carbs. You want to get your carbohydrates broken down into monosaccharides so they can be absorbed in the bloodstream. All of your foods have to be in their smallest units to be absorbed into the bloodstream. Well, why do we need the carbs? What's the main reason we use carbs for? Energy, right? This is all going somewhere. Just be patient with me. All right, so let's say we got glucose. That's a monosaccharide, most common one used in our body. Well, what do we mean by energy? What happens to these monosaccharides, this glucose, this sugar? Well, it circulates around and you uptake it by cells that need that sugar. And what do they need that sugar for? They're going to take that 6-carbon sugar and break it down anaerobically into 2 3-carbon molecules that can go on into what organelle in the cell that will finish the breakdown aerobically. You better be at home thinking mitochondria. By this point in the game, you need to know that. So as the pyruvates go in, which are your 2 3-carbon molecules, breakdown product of glucose, they go into the mitochondria. What is needed to make the aerobic phase of cell respiration work? Oxygen, right? Oxygen. So here now, where's that oxygen come from? We've got to take it in through what system? Respiratory system. Then we got to transport it from the respiratory system through the bloodstream to the cells that need the oxygen. So I'm getting full circle here. This is a metabolic process, right? Cell respiration is a metabolic process. So you get into the mitochondria and you start breaking down your carbon-to-carbon bonds in order to get the energy from there and transfer that energy through the electron transport chain to phosphate bonds. What are you making with phosphate bonds? What is the usable form of energy in our body? What do cells need energy in the form of? ATP primarily, right? Adenosine triphosphate. So you convert that energy over there. Well, in the meantime, when you've broken down all these carbs into waste products to get the energy that was found between the carbon-to-carbon bonds, right? then you're going to breathe out, or the cells are going to excrete a waste. What's the gaseous waste from cell respiration? CO2, right? So CO2, it's going to be excreted, I should say, from those cells, and it's going to be transported in the blood. How's it transported in the blood? All right, you better be thinking this. 80% of it as carbonic acid. What's that going to do to your pH of the blood? It's going to swing it toward the acid side. But if all works well, you're not going to allow it to get below 7.35. There's going to be mechanisms, bicarbons and things that are going to be put out there that are going to buffer that acid to try to keep it in the range. So we have tied together energy, muscle contraction, no, not that, I'm going to get that in a minute, respiration, metabolism, digestion. See the big picture I'm talking about? Let me give you a scenario. You are exercising. Let's say you're running. Let's say you're a runner, and you've been running for an hour. You've been running for an hour, okay? Vigorous exercise over a prolonged period of time. What is required to allow your muscles to contract and run and work that long? What do you have to have? A lot of energy, right? Got to have a lot of energy. Well, how do we get most of that energy? Most of that energy is gotten from the aerobic phase of cell respiration, right? So we need oxygen. We need to get oxygen in. But as we are generating that muscle contraction and as we are generating the ATP, we are also building up a lot of waste of CO2, right? So what would you expect the pH of the blood to do when someone is vigorously exercising for a prolonged period of time? They're going to be producing a lot of what? Ultimately carbonic acid, which is going to do what to the pH? Lower the pH, right? And actually what you need to understand, and we'll get into this probably with the respiratory system a little bit more, but there are cells in your hypothalamus, in your brain, that's going to pick up on this increase in hydrogen ions, because remember, carbonic acid is very unstable in the blood. It quickly dissociates, producing a lot of hydrogen ions. So the hypothalamus is going to pick up on this increase in hydrogen ions or acidity of the blood, and it's going to say, whoa, we got to get rid of this CO2. It sends a signal down to your respiratory center, which is in your medulla oblongata, that says, pick up the pace of the breathing. We got to get rid of that CO2. And in fact, what we know is a stimulant for breathing more rapidly and deeper has to do with the need to get rid of this carbon dioxide waste and the need to keep that blood pH in the normal range. So if you had someone ask you which stimulated the breathing, I don't want to hear you say that you need to breathe to get oxygen. You do, but that's not the main stimulus. The main stimulus for breathing is to get rid of CO2. One of the reasons, yes, we do need oxygen, but one of the reasons CO2 comes before oxygen is because we have all kinds of ways to store oxygen. Do you remember myoglobin in the muscle cells? If you have been regularly exercising and you are demanding that of your body begins to make more myoglobin that has the ability to store oxygen and save it up for when you have these prolonged vigorous exercises and that you need. So we can get oxygen from other places in our body and carry out what we need to do. So the trigger is we're building up CO2 waste. We got to get rid of CO2 waste. Okay, thank you for my digression now. Let's get back to the problem solving, because I wanted you to understand how all these systems of the body are reliant upon having that pH maintained in a normal range. All right, the next slide is going to talk about something called compensation. Compensation is kind of like what I was describing a minute ago. It's the ability of the body to try to stabilize the acid-base balance. And to stabilize it, what we mean is we want to keep it within that 7.35 to 7.45 range for pH. If the pH is in the normal range, but the other two values are out in opposite directions, and I'll explain what that means in just a minute. then the body is compensated for the imbalance. In other words, you're going to have two things that can swing in opposite directions. If the pH is swinging out toward acidosis, you might be able to do something with one of those other ions, the carbonic acid, bicarb ion, or the CO2 ion. You might alter those in order to try to pull the pH back in range. And so if they are out, then the body is basically compensated. Then if the pH is out of the normal range, then you look to see whether the body tried to stabilize, which in biology we call partial compensation, or whether the body didn't try to stabilize that. In that case, we call it uncompensated. So let's go back to give you just one more short rule. Basically, when you're looking at these problems, if the pH is in the range, you can say that it, and the other two might not be, but if the pH is in range, the body has compensated to make sure that you've got the pH in the range. And if the pH is out of range, your body has uncompensated, okay? Has not done the right thing to get it back in. Now, that doesn't mean you're totally out of the woods as far as having a problem. It is simply an indication of a problem, and it tells you where your problem is. So that's one of the important things to look at this. We're going to see that the nursing department only requires you to determine whether the acid-base balance has been compensated or is uncompensated. I'm going to take you a step further because it is important in biology, and it's important for the doctors. So you're going to med school or pharmacy school, you're going to be interested in looking at whether the body attempted to compensate, because that's going to help you with a diagnostic tool. So I'm going to teach you how to see whether the body did what we call partial compensation. But I'm here to tell you that when you hear partial compensation, basically it just means that the body tried, but it didn't work. So really, even partial compensation is an uncompensated situation. All right, don't panic yet. We're going to actually try to do some problems here. All right, so what I want you to do is I want you to get that sheet out that I told you to print off that has the steps, because it gives a scenario. But basically, step one, I didn't put step one on here for some reason. So step one is look at the pH. So the first thing you do is look at the pH. So we're going to take the first case that's in this story. Let's see if I can find it. Example. We're taking Mrs. Norton. Ms. Norton is 60, has a history of ulcers, and was admitted to the hospital where a nasogastric tube was inserted after several days of vomiting. Where'd they put that tube? Down her nose and into her stomach, right? Her lab values for blood chemistry include, she has a pH of 7.52. Is that in range or out? It's out. We'll write it down in just a minute. She has a PaCO2 of 49. Is that in range or out? That's out. She has a bicarb ion that is 40. Is that in or out? That's out too. So here's what we do. I'll come back to this matching value in a second. When you work these problems, since science is a methodical thing, and this is a step-by-step method, I cannot emphasize enough that what you need to do is have your ranges written down. So start with your pH range, 3.5 to, what am I doing? 3.35 to 3.45. This is your pH. All right? And then go ahead and write your PaCO2 range. What's your PaCO2? Your PaCO2 is 35 to 45. Where am I? Yeah. And your bicarb ion, HCO3, is 22 to 26. Now, I'm not putting your units in there, but that's always very important. Don't forget, whenever you're actually looking at values, you need to put the units with them. So this is just to try to get you to see where we're going with this. All right, again, what I have students do, I'm going to change my color here. All right, what I have students do is I always give you the lab values. I don't make you memorize these ranges. The homeostatic ranges are almost always, I don't even think almost is a word I should use, they are always printed on the lab report. So I see no point in you memorizing these homeostatic ranges. Now, nursing school, if they make you memorize it, do so. But if you've worked enough these problems, you shouldn't have to worry. That'll come naturally. But I suggest that you write this, what I'm writing on the thing, every time you do a problem. So what I want to remind you of is that let's first write which way, whether we're going into acidosis or alkalosis, depending upon each of these values, which way they're going. So what did we say? If the pH was lower than 3.35 on the pH scale of 0 to 14, if it's lower, what, 3 point, God, y'all, 7.35. Too early in the morning for me. 7.35 to 7.45. All right, so if the pH range is lower than 7.35, you're going into what? Acidosis. So write that down like that. If it's higher than 7.45, you're going into what? Alkalosis. Okay? Let's look at the next one before we go on. PaCO2. How's that carried in the blood? Carbonic acid. Again, write this down. Write it down every time until you get this method down. If you have a high level of carbonic acid in the blood, higher than 45, you're going into what? Acidosis. If you have a lower than 35 PaCO2, you can go into alkalosis. All right. Now let's look at the buffer, the bicarbonate, your buffer. If you have a lot of buffer, if you have more than 26 milliequivalents, you're going to have the problem of alkalosis. If you will say this to yourself every time, and you will write this to yourself every time, you will not miss these problems. If your buffer is lower than 22, then you could have a possibility of going into acidosis. All right, so now let's go back and look at Ms. Norton's values, and I'm going to go back, and we're going to write them on the scale where they fall. So Ms. Norton has a pH of 7.52, so put it up here, 7.52. So look at your pH first. What does your pH tell you? Your pH tells you that she's going into alkalosis, 7.52. Now, the next step is to find the matching value. Now, what we mean by matching is find out if one of these other values is out in the same direction, and meaning not left and right, meaning acidosis or alkalosis, that her pH is. So let's look at what her other two values are. Her PaCO2 is 49. So again, write it on your chart, 49. The pH indicates she's going into alkalosis. The PaCO2 indicates what? She's into acidosis. Oh my, what do we do with that? That's out that way. So let's look at the bicarbonate. The bicarbonate is 40. She's got a 40 milliequivalents of bicarb. So here's what we mean by matching value. Oh, by the way, the other thing I'd recommend you writing is that your CO2 is your respiratory parameter. Remember that? And your HCO3 is your metabolic parameter. That's important. So write that on there too. So now let's go back. What we want to do is we look at the pH first. It's out toward alkalosis. So you next look at your other two parameters and you find the parameter that matches the pH. Here it is the bicarb ion that's also in alkalosis, which is what her pH is, which means that matches. Right there, it tells us that this person has metabolic, and these two values tell you alkalosis. In fact, the pH itself will tell you whether you're in acidosis or alkalosis. So start right there. The pH says this person is in alkalosis. You look at the other two values to see whether it's a respiratory. Now, let's look to see whether it was compensated or uncompensated. Remember in nursing school, compensated equals pH in normal range. But the other values are out. And uncompensated, it says that the pH is out of range. Okay? So right away we can look up at this problem and say, okay, the pH is out of range, so this lady has uncompensated metabolic alkalosis. All right? Uncompensated. Now I'm going to take it a step further. We're going to look to see whether she tried, whether her body tried. So what you do when you're trying to go this other step further, that again, if you're in nursing, you don't have to do, but I'd like for you to understand it, is you look to see whether the value that didn't match, which would be the respiratory value, whether it attempted to try to bring that pH back in line. If the pH was out on the alkaline side, then you would want that other value that didn't match to go way out in the opposite direction, which is the acid side, and it did. So in biology, we look and we see that the respiratory value is way out on the acidosis side also. So in biology, we would say that this lady was had partial compensation, which means her body tried to correct that pH. Her body swung out one of the values toward the side opposite the pH in an attempt to try to bring that pH back in line. It didn't work, so she's still uncompensated, but her body tried. And again, that's going to be a good diagnostic tool to see whether some of her body is functioning correctly or not. All right, I'm going to grab the eraser here, and what I want you to do is I want you to go ahead while I'm erasing and try to work the next example. The next example is Mr. Long. Mr. Long, I'm reading it right off this thing you should have printed off. Okay, print that off and look at it. Mr. Long has an acute renal tubular necrosis following cardiopulmonary arrest. Boy, this guy had a lot of problems. What happened to his heart and his lungs? They quit working, right? So cardiopulmonary arrest is his heart quit beating and he quit breathing, but they revived him. So now he's got he has acute renal failure, and the failure happens to be in the tubules of the kidney, and you're going to learn those in this unit too on the urinary system. And what does necrosis mean? Tissue death. So what we're saying is he's got tubes in his kidney that are a very important part of the way the kidney functions that is dying. So he's going into renal tubular failure. Blah. All right, so let's look at his values. He's got a blood pH of 7.32, PaCO2 of 37, and HCO3 of 17. So you take your scale, as I'm going to also. You take your scale, and I want you to do exactly what I said to do. Why is it not letting me erase? I'm recording. They're making faces at me. All right, while I'm erasing, you guys should be working this problem. Remember that the first step that you should make is you should make sure you have all your parameters, your homeostatic range here. then really, I can't emphasize this enough, write down and think to yourself, lower than 7.35, you're in acidosis. Higher than 7.45 on the pH scale, excuse me, you're in alkalosis. PaCO2. If you've got a high PaCO2, you're producing a lot of carbonic acid because you're producing a lot of CO2 that changes to carbonic acid and you're going into acidosis. Low PaCO2, lower than 35, alkalosis. Buffer, high buffer, outside of 26 is alkalosis. Lower than 22, acidosis. Write those things down, okay? All right, now I'm going to take and write the values. You should already be there. Hope you're already figuring this out. pH is 7.32. So where do I put the pH of 7.32? Down here. Is it out of range? Yes. Which way is it out of range? It's lower than 7.35, correct? Please. Let me correct this because it's bothering me. It's like I can't believe I stumbled on that. Anyway, all right. So, right away, here's his problem. This is Mr. Long. Yeah, Mr. Long is in acidosis. Okay? You can tell me right now by looking at that pH. Is it compensated or uncompensated? It's uncompensated. So now the problem is we've got to figure out whether it's metabolic or respiratory. So let's remember that the next thing you have to do is match your pH. So let's look at the other two values. Your PaCO2 is 37. Where is it? Is it in range or out of range? It's in range, right? So his carbonic acid levels, his PaCO2 is just fine. Let's look at the other values. His bicarbonate is 17. His bicarbonate is down here below 22. So he's in acidosis, isn't he? Remember what I said. The PaCO2 is your which value? It's your respiratory. Your bicarbonate is your metabolic. So if he's in acidosis and the bicarbonate is the one that matches, it's also in acidosis. What does he have? Metabolic. Acidosis. Nurses, what do you know? Is it compensated or uncompensated? Well, pH is out of range, so what? Uncompensated. Metabolic acidosis. All right? Take a step further. Did the value that did not match, the opposite value, did it attempt to correct the problem? The opposite value is your bicarbonate ion. And did your respiratory functions try to correct it? Well, it would have to be lower than 35, right, to show that it had partial compensated. So this was not even a partial compensation, okay? Not even a partial. So this is just a pure uncompensated metabolic acidosis. All righty, boys and girls. At the bottom of this sheet, you have some practice problems, and I'm going to do 3. I'm going to help you do 3 of them, so you're only going to have 5 left to do when I'm through today. So let's try another one, okay? You try, we're going to go to number 1. So try number 1 while I'm erasing my board. Okay. Number 1 has a pH of 7.31. Again, you fill out your board. You should know everything that you got up there. Put your value where it falls on the homeostatic range. 7.31 is what? This person has acidosis. pH tells you that in acidosis. What's the next step? You find the match. So let's look at the other two values and see. PaCO2 is 50. Write it where it falls. PaCO2 is 50. What about the other one? HCO3 is 22. All right. What's the next step? Find the match. So if this person is in acidosis, the respiratory value is also in acidosis. Notice how it's out of range on the acid side. So this value right here matches the pH. Here's your pH out on the acid side. So this person's problem is what? Respiratory acidosis. Next step, figure out whether it's compensated or uncompensated. How do you do that? You look at the other value. You look at your HCO3. It was not the match, so let's see. Is it in range or out of range? Still in range. So this is an uncompensated respiratory acidosis. Was there partial compensation? No, because if there were, this, where are we? Which one was it? Respiratory, so we're looking at metabolic. If this were partial compensation, you would want that 22 to be out above 26. Have to be out above 26 going in the opposite direction to partially compensate it, but it didn't work. Okay, we're going to do another one. Let's jump to 3. We're going to go to 3. So you look at 3, start all over, put all your pH ranges, your PaCO2 range, your HCO3 range. Think to yourself, write on your ranges, which way's acidosis, which way's alkalosis. Write all that down. Then go back and look at PaCO2 and you say to yourself, that one is the respiratory parameter. Go to HCO3 and put down, that one is the metabolic range. You get all that done while I'm erasing my board. Okay. I'm going to go to 3. Problem 3 has a pH of 7.47. What do you do first? You could say 7.47 is out on alkalosis, so this patient's problem is alkalosis, right? Now let's look at the other values. And listen, what you guys need to do is if you're still working these problems, when I start, pause this darn thing. Pause it, try them for yourself, then start me up again and see if you were getting it. So number 3, the PaCO2 is 48. So he's out up here on the acid side on that parameter. And number 3, the HCO3 is 30. He's out on alkalosis. So, after we've determined by the pH that his main problem is alkalosis, what do? Find the matching value. So, we've looked to see alkalosis here, alkalosis here. So, since this value is out on the alkalosis side, what is his main problem? Metabolic acidosis. Now, what do we do next? We look to see whether it is compensated or uncompensated, correct? Well, you go back up to your pH. You guys in nursing, it's out of range. It's what? Uncompensated, right? But if you want to know more, if you want to see whether or not the body attempted to compensate, see if it's partial compensation, you look at the what? Opposite value. Did the opposite value make an attempt in this case to pull the pH back into range? Sure did. So in biology and in med school, you look at this value and you see that this is out of range in the opposite direction. So we call this partial compensation. Okay? Again, this is all you need to know in nursing. Going any further, partial compensation is important because it's an important diagnostic tool. All right, we're going to do one more that's a little bit different before I quit today, and that's number 7. So I want you to go down to problem number 7. And you may have to let me get you started before you get very far into it, but this is a good time if you'll pause me and set up your homeostatic parameters again and practice going which side's acidosis, which side's alkalosis, which value is respiratory, which value is metabolic. If you'll practice that while I'm erasing, that'll be a good thing. All right. What's different about this one? Number 7. pH is 7.45. This patient looks fine. This patient's pH is in the normal range. Is this patient, by looking at that, does this patient have acidosis or alkalosis? Doesn't look like it. But let's look at the other two values. Which one am I doing? 7. So the PaCO2 is 34. This one's out of range, isn't it? The respiratory value is leaning toward alkalosis. Let's go to the HCO3, and the HCO3 is 20. The HCO3 is also out of range, but it's out of range toward the acid side. So what on earth do we do now? We got a pH in the normal range. One of our parameters is out toward acidosis, the other's out toward alkalosis. So how do we figure this one out? Here's the key. When you have the other values out, you take your pH range and you split it down the middle. So the pH range that's in the middle is 7.4. And what you do with that is you look at your pH, and if the pH value is to the alkalosis side, a higher number than 7.4, but still in range, between 7.4 and 7.5, we consider the problem to be alkalosis. If the patient's pH happens to fall between 7.35 and 7.4, in other words, 7.35, 7.36, 7.37, 7.38, 39, up to 3.4, then this patient is considered to have the acidosis problem. And that's only if these other two parameters are out, because otherwise, if everything's in range, you're what? You're fine, right? But this patient still has a problem. So what I do is I teach you to split the pH down the middle when the other two are out of range, and you match the pH on the side that is closest to. Then you look at the other values for the match. So the respiratory function had a PaCO2 of 34, which is illustrating alkalosis, right? Well, is that not a match? Because your pH is almost out of range, is right on the edge of being out of range into 7.45, so it is in alkalosis, or you can consider this the problem. So because your match here is between your respiratory value and your pH, then this is considered respiratory alkalosis. All right, since this was out. But go back to what I said about figuring compensation and uncompensation. When you look at that range, the pH is in range. So the other value's out in the opposite direction, right? So did it compensate? Absolutely. This metabolic phase, the bicarbonate was kept or prevented from being secreted to the point where your bicarbonate was low, 20 milliequivalents. It only had 20, so it was leaning toward the acidosis side, and that in itself pulled your pH back into range, which made this a compensated respiratory alkalosis. Okay? Then you might say, well, what if you get a pH of 7.4? And my answer to that is, that's what you pay the doctors the big bucks for. They go to med school to figure out what else they have to look at besides that. So your job now is to go back and practice these problems. Try to do the ones that I didn't do. You have answers also available to you in the case study folder. But you know, it's like math. If you don't try them yourself, if you just try to look at the answer, memorize these particular ones, your ship is sunk. I'm going to tell you that, because you're not going to have a problem exactly like these. But they use this step method. If you know how to do this method, if you practice this method, you'll be fine. you can even make up values. That's what I did. These practice problems, I just made them up. So you can do the same thing. Make them up. Make as many of them as you can and see if you can figure out what the problem is. Good luck. If you have problems, let me know. Have a good day. Bye.